Hello everybody. After hatching from the zona pellucida, blastocyst is traveling through the fallopian tube into the uterine cavity, where it implants into the mucosa of the uterus called endometrium. We already know that the cells on the surface are called, or form a layer called trophoblast. The leading front of this trophoblast starts to form a multinuclear layer called syncytium, which invades into the which invades the endometrium by releasing proteolytic enzymes. Inside there is the embryoblast layer, the inner cell mass, where the embryo will develop. So let us say we are looking on the implantation on day 6. The implantation has started recently, and we will see the glands of the endometrium in the secretory phase, therefore highly tortuous. The endometrium is lined, as well as the glands, they are lined with simple columnar epithelium. The glands contain some secretion because it's the secretory phase of the endometrial cycle. The endometrium contains blood vessels which are also invaded by the Syncytiotrophoblast. Okay, here's another gland. We should not forget the endometrial stroma, which is the connective tissue component of the uterine mucosa. So the cells on the surface, well, I will add the nuclei. The cells on the surface form a layer called trophoblast. The trophoblast on the invading pole forms a multinucleate syncytium, therefore Syncytiotrophoblast, that produces hydrolytic enzymes helping to invade. Well, the first step of this invasion was preceded by adhesion, mediated by adhesion molecules stimulated by cytokines of the endometrium. Inside we have the cavity of the blastocyst. The cavity is called blastocell. And we have the embryoblast. Which is usually oriented on this pole that is the leading pole of the invasion. We got the glands of the endometrium. With some secretion course, it's the secretory phase. Below the simple columnar epithelium, we got the connective tissue stroma. The connective tissue component of the mucosa, and we got the uterine blood vessels. Or the endometrial branches of the uterine blood vessels. So that's how it looks like on the day 6 approximately. The Syncytiotrophoblast is providing nutrition to the growing blastocyst by decomposing the nutrients that have been stored in the endometrial cells. The endometrial cells at this time are filled with lipid droplets and with glycogen. A few days later, the implantation proceeds, and something happens here within the embryoblast. So we still have the cytotrophoblast on the surface. It has a shape of simple cuboidal epithelium. We got the multinucleate syncytium that forms this kind of villi that invade the endometrium. So there have been mitosis of the cell nuclei, but no cell division of the daughter cells. That's how we got the syncytium. And this embryoblast formed two vesicles. One that is called yolk vesicle, and it's lined with cells called hypoblast, and another vesicle called amniotic vesicle, lined with cells called epiblast. So let's add the nuclei. Both vesicles are embedded in a primitive connective tissue called extraembryonic mesenchyme. Therefore, I'm adding spindle-shaped or star-shaped connective tissue cells or mesenchymal cells. There are cavities within this connective tissue that will gradually fuse. And I add some of the extracellular matrix here. And again, we got the uterine glands. Of irregular shape. And the blood vessels, some of which are even eroded to form blood lacunes in the space among the villi. The blood supply is especially rich in this phase of the endometrial cycle. And of course I will add the connective tissue stroma, but it remains the same like on the previous scheme. So what is new here? This is like 2 or 3 days later. So day 8 to 9 after implantation, we still got the trophoblast on the surface. However, inside the embryoblast, 2 vesicles emerged. Namely the yolk vesicle which is lined with cells called hypoblast, and the amniotic vesicle, lined with cells called, or layer of cells called epiblast. They both embedded within undifferentiated connective tissue called extraembryonic mesenchyme or primary mesenchyme. Extraembryonic means it's outside of the future body of the embryo, which will emerge on the contact between the yolk and amniotic vesicle later on. There are some lacunes inside the extraembryonic mesenchyme. These lacunes will gradually fuse to form the extraembryonic coelom. Coelom, that's the cavity, C-O-E-L-O-M. And we got the endometrial blood vessels and the glands. The implantation starts on day 6, which fits the so-called implantation window That's the time interval when the endometrium is receptive. It's prepared and able to implant the blastocyst. This implantation window ranges between the 20 to 23 of the menstrual cycle. The menstrual cycle refers to the cyclic changes of the endometrium. And if you consider that the ovulation occurred at day 0 of the life of the new embryo, that was the day 14 of the menstrual cycle. Let us say the fertilization occurred the same day. So 6 days after ovulation overlaps with the 20th day of the menstrual cycle, right? So that's where it starts. The whole implantation takes approximately 5 to 6 days. So let's say on day 11 it's completed. So you got the renewed epithelium that has closed above the implantation site. Well, there's some fibrin plaque for some time before it's overgrown by the simple columnar epithelium of the endometrium again. But the whole embryo is now fully implanted. Now the Syncytiotrophoblast, which was here, is overgrowing the trophoblast. Therefore, we need to distinguish 2 layers of the trophoblast now. It's the one that has this form of simple cuboidal epithelium. Let's call it the cytotrophoblast from now on. Okay, 1 layer of cuboidal cells. Will persist through the first trimester. Now it's surrounded by the Syncytiotrophoblast, but on this pole it does not form any villi. However, where the implantation started, there's an extensive growth and branching pattern of villi invading the endometrium. So I will add multiple nuclei into this basophilic layer called Syncytiotrophoblast. The legumes of the legumes inside the extraembryonic mesenchyma have fused into a large cavity. Therefore, the extraembryonic mesenchyma was pushed to the periphery. So it's on the periphery. And here it connects the two vesicles to the periphery. So we already know we got the yolk sac that forms kind of a finger-like protrusion here, and we got the amniotic sac. Both are in close touch and they will interact later on to form the embryo. So here's the extraembryonic mesenchyma. We got also the surrounding glands of the uterus, of the endometrium here. With their simple columnar epithelium. We got the connective tissue stroma of the endometrial mucosa. And we got the blood vessels, some of which form kind of blood legumes here in the space among the villi. And they will later on establish the placenta circulation, yeah, the maternal component of the placenta circulation. Another word for the endometrium in pregnancy is decidua. This is the capsular decidua, the one that covers the implantation site. So decidua capsularis. This is the endometrium, endometrial epithelium. These are the glands. This is the stroma. Here there is a temporary fibrin plaque before the epithelium renews its integrity. And if you consider these three layers, namely the Syncytiotrophoblast, the below lying Cytotrophoblast, and the extraembryonic mesenchyma, they together form the outer fetal membrane called chorion. Now in the region where chorion forms no villi, it's called the smooth chorion. Chorion levi in Latin, leve, that means smooth if you would translate it, without villi. But in this region it's the chorion with the villi. So here you got so-called chorion frondosum, which means with the villi. Okay, here we got the uterine blood vessels. And blood lacunes. And the two vesicles, the yolk vesicle with this finger-like protrusion called allantois, and this vesicle, which is the amniotic. They both connected to the wall via this connecting stalk. Let's label it like here. Yeah, that's this region. And the cavity here is the extraembryonic coelom, which is different from the intraembryonic coelom that will develop later on, as we'll discuss it after gastrulation. Now let's pay attention to this region where the amniotic and the yolk vesicles are touching each other. That's the region of the so-called bilaminar embryonic disc. Bilaminar means it consists of two layers, which are the lining of the amniotic vesicle, that's the epiblast from the amniotic vesicle, and the lining of the yolk vesicle, which is the hypoblast from the yolk vesicle. And we will describe the process of transformation of the bilaminar embryonic disc into a three-layered gastrula, into trilaminar gastrula, which will consist of the following three layers called embryonic germ layers: the outer ectoderm, the middle mesoderm, and the inner endoderm. This process is called gastrulation, and it will be covered on separate schemes. One note to the Syncytiotrophoblast here: it's a huge mass of tissue that is producing, among others, a hormone that is so specific for pregnancy that it's used as a biochemical pregnancy test. The name of this is HCG, which stands for human chorionic gonadotropin. So this could be detected in the urine or in the plasma, and its levels, the increased levels or its presence detects the presence of the Syncytiotrophoblast in the female body. Let me add two terms here to describe the villi of the Syncytiotrophoblast. And if I mention the decidua capsularis on this pole, then the decidua here basally is the decidua basalis. It's the basal plate of the endometrium below the site of implantation.